Good afternoon and welcome to an EAS webinar on emerging pathogens presented by EAS independent consultants, Drs. Kathy Knudsen and Kathy Glass. EAS Consulting Group is a member of the Certified Family of Companies and is a global leader in regulatory solutions for industries regulated by USDA, FDA, and other federal and state agencies. Our network of over 150 independent advisors and consultants enables EAS to provide comprehensive consulting, training, and auditing services, ensuring proactive regulatory compliance. Dr. Kathy Knudsen is a microbiologist and certified lead instructor for the preventive controls for qualified individuals through the Food Safety Preventive Controls Alliance. She consults with companies in meeting FSMA requirements, including manufacturers in the cannabis industry. She has a PhD in food science from the University of Minnesota and is the author of Food Safety Lessons for Cannabis-Infused Edibles. Dr. Kathy Glass is a distinguished scientist at the University of Wisconsin at Madison, where she directs the Applied Food Microbiology Laboratory, including developing research protocols and supervising projects relating to the evaluation of microbiological safety of low-acid, refrigerated, and formulation-safe, shelf-stable foods. She is also a process authority for evaluation on formulation-safe foods. As a reminder, during the webinar, you may type any questions by typing them into the questions box. And with that, I'll turn the presentation over to Dr. Kathy Knudsen.
Thank you, Amy. So you're going to get a 1-2 punch here from Kathy and Kathy about pathogenic E. coli. My presentation is going to be more on the higher level view, just touching really quickly on a lot of different topics related to E. coli. And then Kathy, in her expertise, is going to be showing you some data and looking at more specific information. Let me get my slides going. Oh, it did it. Yes, we were anticipating that. All part of the plan here. There we go. So you should have the next slide coming up, which is objectives. I'll go through these briefly with you. Define Escherichia as a genus within Enterobacteriaceae in the coliform group. Explain the importance of indicator organisms in an environmental monitoring program. Define pathogenic E. coli, Escherichia coli. Demonstrate hazard identification of pathogenic E. coli using the FDA Appendix 1 guidance document, and summarize FDA Leafy Greens STEC action plan. So as you think about that, big group of information there. One of the reasons that our topic is so important, and I'm so happy to be collaborating and working with Kathy Glass on this, I had a client who found generic or non-pathogenic E. coli in their product and then on a food contact surface. And their FDA 483 questioned the company as to had they investigated whether there was pathogenic E. coli present. And this was the first time in my career, this was a couple of years ago, first time in my career that I have seen this leap from a discussion of generic, what I call generic E. coli, to pathogenic E. coli. So let's explore, starting with the introduction to Escherichia coli, which we all, of course, just call E. coli. Most E. coli do not cause illness. Most are harmless, do not cause harm. They are gram-negative rods. They are facultative anaerobes. And so just thinking back to our general microbiology days, we all learned a lot about E. coli in that regard as being not harmful, not being a pathogen. The picture that I'm showing you is my 2 little joy bombs that were born last year. I have 2 grandsons, and so they have brought a lot more fecal matter into the household as we care for them. So what are the sources of E. coli? E. coli is naturally present in the intestinal tracts or the intestines of animals without any harm. And so that is true for us. humans. That is true for cattle, birds in the air, and then the bird droppings coming down from the air, the wild animals roaming through our fields, the mammals that are roaming through our fields like rodents and foxes, and out in California, the concern with mule deer in the romaine fields or the spinach fields. The fecal contamination is going to then go from that primary source of the intestinal tract of an animal to plants that are grown outdoors, soil, water, and think about manure. Manure is fecal matter, and E. coli is going to be there. So fecal contamination is a term that we talk about. Again, going back to that original source, it is the fecal matter coming from the intestinal tract of an animal. But where is that fecal contamination naturally present? So that's going into the water sources. And if that is an untreated water source, there could be the potential of fecal matter and the E. coli that's in that fecal matter to still be there. And that's why we treat our water for sanitary water. Again, the soil can be from the bird droppings, it can be from the animal droppings, all that being naturally part of animals and their habitats. Then if we think about raw animal products, the outside of eggshells, ground beef, in that when cattle are butchered, that there is the transfer of the fecal matter from the hide of the animal to the beef, and then getting all mixed up in our ground beef. That's why we have to cook our hamburgers all the way through. Pieces of raw chicken are known to have fecal matter, fecal contamination. Raw milk is known to have fecal contamination as well. And again, those crops that are grown outdoors. So I just want you to think through this whole process of starting with the intestinal tract of an animal, the fecal matter, the fecal contamination, which we all need to remember is absolutely natural. It's naturally present in the water sources, our soil, raw animal products, and crops that are grown outdoors. So let me tell you a quick story. As Amy mentioned, I published a book, Food Safety Lessons for Cannabis-Infused Edibles. I have transferred my from traditional food science and food safety over to the cannabis industry. And I had a question from a hemp processor about coliforms. And the driver was that they had a customer that was asking them to test the incoming hemp for coliforms. And so for me, it was like, okay, time out, wait a minute. Hemp is grown outdoors in fields. It is a plant material. There is going to be naturally present fecal contamination. That hemp is then harvested and brought to an extraction facility. So at receiving, there's going to be coliforms, there's going to be fecal contamination. It goes through a grinder, and then the ground plant material goes to extraction. And that extraction typically is a very harsh chemical extraction, and you would not expect the fecal contamination to survive through that process. But it absolutely does not make any sense to test that incoming hemp for coliforms, because number one, you're going to find it. And then number two, what's your corrective action? What are you going to do about it? And in the case of growing hemp, there's nothing that you can do about it. It is naturally present. So fecal contamination, thinking about that as a concept, we now are going to move to inside of our facilities and think about the term of sanitation, or in the case of finding fecal matter or fecal contamination, I shouldn't even say that. In the case of finding coliforms, it could be considered poor sanitation. So what is the source of contamination when you do find coliforms within a facility or E. coli within a facility? Again, going back to that original source of the intestinal tract of an animal, the fecal matter then being transferred onto or into water, soil, an animal product, a raw animal product onto a crop, or those humans that come in the door of our facility as well. And they could be bringing the contamination in with them. They're certainly bringing fecal matter in with them because that's perfectly normal. Transferred throughout the facility by the traffic patterns of those people, those humans that get to come into your facility and walk through your facility, and all of the wheeled equipment. So whether you have forklifts, hand trucks, carts, other types of wheeled equipment, they could be transferring from receiving, where it may be perfectly normal and natural to have contamination, but then it could be brought into your facility. And how are you going to control that? You're going to control that through your GMPs and through good sanitation practices. This is one of my favorite figures in the whole world because I'm a geek and a microbiologist, and this is the family Enterobacteriaceae, which is really fun to say. And if it's hard for you to say it, my advice always as a microbiologist is just say it really fast. And people don't know if you know how to pronounce it correctly or not. But this diagram comes from a wonderful document from ILSI. Down on the bottom is the citation. International Life Sciences Institute has an article called The Enterobacteriaceae and Their Significance to the Food Industry, which is a publicly available PDF. And among the members of this family, we have many genera, lots and lots and lots, more than I have ever heard of in my life. And many of them are non-pathogens. Some of them are pathogens. And so our non-pathogens are commonly found in nature, normally present, non-pathogenic, and would be expected to be found in nature. So in our natural waters, in soil, on plants. And then when you think of pathogens, when I think of pathogens, I think of the intestinal tract and fecal matter. So the way that you read this diagram, if you start on the very bottom, and Amy and Kathy told me I can use my pointer down here. So the outside genera, the genera that are in the outside circle area, majority do not ferment lactose. And then if you go to B, variable or slow lactose fermentation. So for example, we have Salmonella, where some crossover, we have Shigella, where some crossover. And then in the smallest, with the smallest number of genera, we have the coliforms. And the coliforms, the majority ferment lactose rapidly. And of course, we use that to our advantage in the laboratory to be able to find coliforms. So we have Klebsiella. There are pathogenic Klebsiella pneumoniae, causes pneumonia. Enterobacter, naturally present out in nature on plants. If I were to have to go and find you some Enterobacter, I would just go get some plant material. Our Escherichia is here, Citrobacter is here, Serratia is here. And this red triangle, which you're going to see again on another slide, is outside of the circle of coliforms and represents pathogenic E. coli. So the majority of our non-pathogenic Escherichia are in the circle and defined as being a coliform. Our pathogenic E. coli are outside of the circle and not considered to be coliforms. And primarily that is because of the testing procedures. Those are completely different tests. Non-pathogenic E. coli or pathogenic E. coli versus coliform testing, which would be for non-pathogenic E. coli primarily. Okay, so here's the enlargement. So with our indicator microorganisms, there are choices in methods, and working either with your internal lab or your external lab, you can choose Enterobacteriaceae count, which is commonly called EB, coliform count, E. coli count. We're going to look at this a little bit more, but know that if you are getting an E. coli count, you can get that on, you can do a swab of a food contact surface, you can do flume water, for example, you can do product. I would not recommend doing that, but as far as different types of samples that you can do. The reason that I say that I would not recommend doing product is just as a consultant, my perspective is that I like to do a lot of building in of food safety. So I recommend that my clients do a lot of food contact surface swabs, and that would minimize your finished product testing program. So then if you're doing an EC count, that literally is just for Escherichia, for E. coli. And it's very, very limiting compared to our coliform count, which has this handful of genera, and it's going to open up and be able to bring in and detect more genera within the family, but still define them as coliforms, compared to the previous diagram, which was the whole family. And if you do an EB count, you are going to potentially get a higher number and potentially detect a greater number of genera than just the small handful that are in the coliform group. So let me go to my points here. EB count is highly recommended for that reason. It's more inclusive. You get a better picture of the sanitation of a food contact surface specifically. So in an environmental monitoring program for food contact surfaces, reporting of either one of these, EB count, coliform count, or E. coli count of greater than 10, that is detectable. So detecting from a clean site, from a swab, would be an indicator of poor sanitation, would require corrective action at your facility, and perhaps a review of GMPs, perhaps a review of sanitation program, but it is definitely an indicator of poor sanitation. Okay, let me go on to my next concept here. Just looking at some of our other indicator tests, I want to emphasize to you that microbial indicators are not pathogens. And this goes to my comment previously, which is that testing for pathogenic E. coli is a very specific test. It is completely different than these indicator methods. So many of us on our swab from food contact surfaces are doing aerobic plate count. This is a general number of bacteria present, including both gram-negative and gram-positive, but I would expect our generic E. coli to grow in an aerobic plate count, so there is some correlation there. Then there are the three tests that I previously mentioned, the EB count, the coliform count, and the E. coli count. First of all, the EB count is that family, so you're going to get potentially a higher number. It's going to be a better, in my opinion, as a consultant, a better picture of what's going on with sanitation. So it does include generic E. coli in that test as well. And then we have the coliform count, which also includes generic E. coli. And this is the group that is naturally present in our untreated water, soil, plant material, those kinds of things that might be coming into your facility. It is a very well-established method. Most of us are doing coliform count because historically it's been relied on. My preference is the EB just because it's a bigger number and better picture. And then we come to the Fecal Coliform Mpn, which is used for water testing, or E. coli count, which can be done on plates or with Petri film. And those are going to include E. coli because that's the whole point. The whole point of that last test is to determine very specifically, is this E. coli? And the thought there is that the closer you get to understanding whether E. coli is present, the closer you get to that original fecal matter. And it's a very, very narrow test. And so to that point, and I also want to make one more point before I tell you this little story, because I consulted with our consultant, Kathy Glass, and talked this over with her because I wasn't sure about the methods. And that is, pathogenic E. coli may or may not grow in that last category of the Fecal Coliform Mpn or an E. coli count, because it's going to be dependent on the temperature, it's going to be dependent on the medium, it may even be dependent on your product and your matrix. So one quick little story. I had a client that was processing a fruit that was then going into little bags, and it was for one of our big box vendors, and the vendor or the customer had required them to do E. coli, I believe it was Mpn, but it was E. coli testing every 15 minutes. And it was either the flume water that they were testing or the product. I think it was the product, but I don't remember. And so they had literally thousands of these E. coli tests that they had done historically over the last few years, that they were required to do it. And not once had they gotten any count. There had never been any E. coli detected. And so that speaks to my point of, this is a very, very narrow test, and you want to be really careful about using it or not using it because of what those results can tell you. And you might miss it. You just might not be looking for the right thing. Okay, so can I test for indicator microorganisms on site at my facility? Yes. In general, so I just was talking about pathogenic E. coli potentially growing, may or may not grow in an E. coli method. But for the other methods, the aerobic plate count, the Enterobacteriaceae count, and the coliform count, in general, we consider that pathogenic E. coli is unable to grow within the conditions for those tests because of the nutrients provided in the media, because of the time and the temperature. The pathogenic E. coli just needs very special conditions in order for us to find it. And those generally are not met in the first three indicator methods that I discussed with you. Therefore, you can do this testing on site at your facility, or you can send it to your contract laboratory. Importance of microbial indicators in environmental monitoring plan. I am a huge proponent of food contact surfaces being swabbed and tested for microbial indicators. So this is in addition to your visual check. So you want to do a visual observation after cleaning. You want to make sure that that everything just looks visually clean. You are probably doing ATP testing of those surfaces. That is real-time testing, extremely valuable. But our microbial indicators also add to the picture of what is the efficacy of sanitation. So for sure, if you are producing a ready-to-eat product, because of our FSMA FDA rule, you need to have a preventive control for that sanitation process. and most importantly for environmental control as well. So you need an environmental monitoring program to make sure that your finished product has not become contaminated from the environment. And we use our microbial indicators for food contact surfaces in that environmental monitoring program without testing directly for the pathogen of concern. By increasing the amount of microbial indicator testing that you're doing on food contact surfaces, you can minimize the amount of pathogen testing that you're doing from food contact surfaces. Okay. By increasing the microbial indicator testing, you can have a robust environmental monitoring program. And I recently learned, you may already know this, but I recently learned that Canada GAP for fresh fruits and vegetables requires environmental monitoring for their produce facilities, in addition to what we have here in the States. So now we get to pathogenic E. coli. Just a reminder that generic E. coli is of fecal origin. Detecting generic E. coli is an indicator of poor sanitation within our facilities. And so the whole purpose of the previous slides was to take you through that thought process of where is it natural to have E. coli, generic E. coli, versus now when I start to find it within my facility, how did it get there and how many steps removed is it from that original source of the intestinal tract? So then, just like generic E. coli, pathogenic E. coli is of fecal origin, but it's now of an animal that is ill with the infection from the E. coli or harboring the pathogenic E. coli within the intestine. Detecting pathogenic E. coli is done at an outside testing laboratory. So you must contract with a laboratory to do the pathogenic E. coli testing. You can never do that in-house because you have to have cultures as controls, and you would never want to do that in-house. So work with your outside testing laboratory. It is very expensive compared to our microbial indicator tests. It takes more time than an indicator test to get to a result. It is highly specific. So in the way that I've been kind of narrowing down these groups for you, you have the whole family, which is Enterobacteriaceae, then you have the coliform group, and then you have E. coli, and within, or you have Escherichia as the genus, and then within Escherichia, you have serotypes or strains of E. coli that are pathogenic. And that's what's represented by my red triangle in the diagram. Even if you get a pathogenic E. coli test come back confirmed, that particular strain of E. coli may or may not be virulent. And I'll give you a little more information on that right here. So in laboratory speaking, those of you who have been involved in laboratory operations and remembering back to your general micro courses that we first all learned about, it is all about biochemical characterization in order to be able to separate out either Escherichia or E. coli itself. It is all based on biochemical reactions. Okay. Then we had, I was working in a testing laboratory in 1999 when we had PCR brought into the facility. So over 20 years now, we've been doing PCR testing, and that was first developed for Salmonella, Listeria, and now we have that available for pathogenic E. coli as well. So there's advantages and disadvantages, limits for that PCR testing. Serotyping is traditionally what has been done to classify or characterize a pathogenic E. coli, and I will talk a little bit more about that. And then we get to the potential now of targeting and testing for the genes for virulence. So if you were to ever think that you have isolated a pathogenic E. coli, there are laboratories that can determine if the genetic material contains the genes that really are involved in illness determining. And finally, there are different strains of each serotype. And for example, with one of the romaine outbreaks and the investigation, there was the outbreak strain, which was isolated from patients and was isolated from the romaine, but there were additional strains of pathogenic E. coli, E. coli 0157H7, that were found when they started to do the trace back to the farms and the conditions there. So those were determined by whole genome sequencing. So whole genome sequencing is out there and available as well. Okay, microbiologists love acronyms. So just very quickly, in addition to STECs that most of us are familiar with, there are 5 different pathotypes of E. coli, and there, I believe it took this from the CDC. I don't see my citation, but CDC has a really good discussion of pathogenic E. coli. The STECs are part of the group that is EHEC, enterohemorrhagic E. coli, and those are the ones that we are more commonly used to seeing, but just know that there are so many more out there potentially. Then when we look at serotype, pathogenic E. coli is defined by the O antigen, which is the outside somatic part of the cell wall, and the H antigen, which is the flagella. And then it is that combination of O antigen and H antigen that gives us the serotype. And that's one significant number there on my last bullet, 200 serotypes. And there's my CDC. Oh no, that's just for the picture. That's just the picture credit there, which I got off of Wikipedia. So E. coli pathogenicity is defined by genes. And this is an area that I know the least about, so I have the most words, and I'm gonna carry on because I wanna get to Kathy's presentation, and I've been talking for quite a while now. If you are a PCQI, you are familiar with the FDA Appendix 1 guidance document. There is a section on biological hazards, which is what you see here, chemical hazards, and process hazards. And if you read across on the first row, one of the most helpful columns is actually the last column, which is examples. And this is where you see leafy greens. And an FDA inspector can go into a leafy green facility that has fresh cut, refrigerated leafy greens, and they can ask them if they have controlled all of these hazards. And after Clostridium botulinum, which would be for modified atmosphere packaging, we come to pathogenic E. coli, Salmonella, Listeria, and so on. Very often in this document, pathogenic E. coli and Salmonella are both marked. It's very, very common. And they're highly related. You saw that on the diagram. So if I control for Salmonella, do I control for pathogenic E. coli? A good consultant and a good scientist will say it depends, because it does. It depends on so many things. But I do want you to consider this group of pathogenic E. coli, Salmonella, Shigella, when you are thinking about your raw ingredients, and then if you have a kill step, when you are thinking about your finished product, when you are thinking about the environment that a ready-to-eat product, like a leafy green product would be manufactured in. And this leads us to the recent webinar that was held by FDA. July 30th, there was a webinar, I guess that was a month ago now. FDA Leafy Greens STEC Action Plan is out there. March 5th, They published the document, and on July 30th, they had a webinar. There's a webinar recording, and this is an excellent example of a case study. It's more than a case study because there's so much work going on that FDA is involved with in addition to others. And FDA in the action plan has categorized the work and the 3 key areas of focus as prevention, response, and then filling in our knowledge gaps with collaboration of all these different federal agencies, academia, and industry working together. And because it is truly a farm to fork problem, it has to be tackled with everyone who's an expert in their area. One thing that I do want to point out is in the lower left-hand corner down here under EPA, Environmental Protection Agency, there is a call for chemical companies to register their product with the EPA and to create antimicrobial water agents or treatment agents that are going to be used in irrigation of these crops in leafy green fields. And that's new. That is new, and definitely an opportunity for industry out there. Then my colleague, Jeff Kornacki, has an article called Challenging Common Aztec Assumptions in Food Safety Magazine in the current issue. And I am not going to read through these with you, but I refer you to that article to really stimulate our thinking. And what do we know and what do we need to know? In summary, most E. coli are harmlessly coexisting in the intestinal tract of animals. Testing for microbial indicators is recommended to evaluate the effectiveness of sanitation within our facilities. The pathogen E. coli 0157H7 emerged as a pathogen of concern in food in the early 1980s. 40 years later, the industry continues the battle against foodborne illness from Aztecs and 5 other pathotypes of E. coli. So a commodity that is at great risk is fresh fruits and vegetables, including leafy greens. Watch for greater focus by the FDA on this category, and watch for greater method development for the detection of pathogenic E. coli. And my conclusion or strong recommendation for you is to create a strong team of microbiologists to control the hazards, such as that grouping of pathogenic E. coli, salmonella, or Shigella, if that applies to your product. Resources are here. You'll have access to those. Most of those are FDA resources, publicly available. And Kathy Glass is on deck here.
Thanks so much. I'm going to be picking up where Kathy had left off, where she was primarily talking about environmental controls and testing. What I'm going to be taking a look as the next version is not necessarily as STEC as an emerging pathogen, because we know that over the last 40 years that it's been around. What we're really looking at is trying to identify what is going to be more of the emerging behavior and where we can find them in different types of foods. So we're going to be taking a look more at the risks, the behavior, and intervention strategies in food products. A lot of the work that I have really has derived from our work at the Food Research Institute at the University of Wisconsin-Madison. It was founded in 1946 with a mission of doing not only basic research, but also applied research. So what we've learned on the basic side about Aztecs, we then can put in a real food product. So we work directly with the food industries and try to do a hazard analysis, and specifically we're looking at validation or food safety studies. So some of the work that I'm going to be presenting comes from those particular studies. A lot of our work is formulation safety safety, but also we might be looking at cooking. Besides having the microbial sciences building being the source of having a lot of work, we are now starting the new meat science and animal biologics discovery building, which will be opening up in a couple of weeks. And that particular facility will have meat processing equipment so that we once again can mimic what is going to happen in real world conditions with STEC, Salmonella, Listeria, Perfringens, and all those other ones. What we're going to be talking about today is STEC as being essentially an emerged pathogen, but we're looking at new food vehicles. Why we really need to have a zero tolerance. There's a lot of controversy that goes along with it, but it really is going to boil down to the infectious dose that's associated with this pathogen. We'll look at STECs not only in foods in the United States, but what other kinds of foods from around the world are going to be attributed to causing illness, what are going to be our strategies for control, and finally, giving you some examples of how we can validate lethality. We're talking about emerging pathogens. We have to think about what kind of pathogens were available to us or known to us up through the mid-1950s. And when the Food Research Institute still was first in existence, we had the typhoid salmonella, the non-typhi salmonella, as well as toxin-producing Staph aureus and Clostridium botulinum. We did see, however, Shigella, and Shigella obviously is going to be the source of the gene for the Shiga toxin-producing E. coli. Since the 1980s, we have a much greater broad experiences with different types of foodborne pathogens that are not only bacteria, but include viruses, parasites, and toxins that can come from algae. One of the more recent ones will be the E. coli 0157, as well as the other STECs, and Salmonella, Listeria, and Campylobacter. If I'm going to be looking at a timeline of what are significant events or outbreaks associated with this particular group of pathogens, we have to go back to the early 1980s and possibly even before that when they were first able to isolate some sorbitol negative E. coli that were associated with illness. But one of the largest outbreaks that would really put it on the map was back in 1982 when two separate stores of fast food restaurants had outbreaks of E. coli 157 associated with undercooked hamburgers. This then resulted in a lot of work into research and trying to understand the virulence of E. coli 15787, as well as developing cooking recommendations that individuals could take back to their stores to inactivate the E. coli and make the food product safe. In spite of that understanding, in '93, we had another outbreak associated with fast food hamburgers. And this was resulting not because they didn't know how much lethality that they needed to have, but because there were some shortcuts in which the undercooked hamburgers were served to the customers. After this point, E. coli 15787 was declared an adulterant in ground beef because of the understanding that people were used to having undercooked hamburgers, and that because the E. coli that might be on the outside would be ground up and put onto the inside, we needed it to make sure that the inside was going to be properly cooked. So the best way for them to address that was making sure there was no E. coli in there to begin with. So it was really an exclusion rather than focusing really on the lethality. Now, the next year, we had an outbreak associated with fermented dried sausage. At that particular time, the sausage was made with raw meat, went through the fermentation process and drying process without really a cooking process. Because we found out that the E. coli 157, being an acid-tolerant pathogen, would be able to survive long periods of time, it was deemed that they needed to have some degree of heat step that was added to this. And as a result, the Blue Ribbon Task Force was able to support a lot of research in which we could identify how much time, temperature, what kind of pH, what kind of drying was going to be necessary in order to get a 5-log reduction in this particular food product. A couple years later, an outbreak of E. coli 15787 in apple juice, and this precipitated the juice HACCP regulation. Once again, looking at a 5-log reduction of E. coli 157 in this particular food product. 10 years later, another watershed moment with an outbreak of E. coli in spinach. However, spinach doesn't necessarily have an option of having a good kill step. And therefore, the research was going to be based more on trying to avoid having it contaminated the spinach in the first place, rather than looking at inactivation. And in 2011, there was a realization that other E. coli other than 015787 can still cause illness. And after this particular point, the Big Six was then declared adulterants in ground beef. Why do we need to have a zero tolerance for Shiga toxin-producing E. coli? In the United States, about 250,000 individuals will become sick with STEC infections. And of those, about 3,600 hospitalizations and 30 deaths. E. coli 15787 is actually only part or a portion of those particular illnesses. Only 35% of those illnesses are attributed to that particular serotype. The rest belong to the non-015787. Why should we be so concerned about this? And it really boils down to low infectious dose. Not all of the E. coli's are going to have the same infectious dose, but for those that are most highly infectious, as low as 10 cells can cause illness. So when you think about having a highly contaminated hamburger, and even if you do the cooking, you really have to make sure that cooking is thorough in order to eliminate those cells. So growth inhibition in food is not necessarily going to be sufficient. We need to have a lethality step that goes along with this. All ages can become infected. In the more mild cases, it might be bloody diarrhea and fever, but in the more severe cases, 5 to 10% will develop hemolytic uremic syndrome, which potentially can cause permanent damage to kidneys, and it can cause death. The individuals that are young, such as small children under the age of 5, as well as older adults greater than 65 years old who have a weakened immune system, will develop the more severe illness. In Europe, about 1/3 of the cases are children. Which serotypes are the most common? What we're looking at is primarily the Big Seven. And as I said, from the last 40 years, we've been focusing on E. coli 015787. But the other main serotypes of concern are going to be 026, 045, 103, 111, 121, and 145. In 2011, Europe experienced a very large outbreak of 4,000 individuals and 16 countries of 0104H4. This particular pathogen ended up resulting in about 30% of individuals came down with HUS. Why was this particular strain so much more virulent than the other strains, which only had about 5 to 10%? And that was because it was expected that it had characteristics of the enterobacteriaceae E. coli, as well as the Shiga toxin-producing E. coli. And this is the reason why it caused so much havoc. All STECs are not created equal. First of all, they don't look the same if you're putting them on selective differential augers, but also individual strains are going to have differences in acid tolerance as well as salt tolerance. And 0157 is known very much for its acid tolerance. It also has differences in severity of illness. Once again, 0157, which is our main target microorganism for the last 40 years, as well as 026 are the strains that are most associated with HUS as well as kidney problems. However, they are going to be similar in ecology. Primarily, they're going to be in land animals, the ruminants, cattle, deer, but also pigs and some birds. So because they're a part of the fecal contamination, they can be found in water, in fields, and as a result, in raw produce, raw milk, as well as raw meat. Besides the outbreaks, we can take a look at case control studies to determine what are going to be some food items that are associated with sporadic STEC infections. This was from a publication from the WHO in 2019 that demonstrated that yes, beef and meat are going to be still on the top of the list, but as well as produce, and also a big player is going to be dairy. In the United States, many of our dairy products are going to be made with pasteurized products, pasteurized milk. However, in places other than the United States, and even a growing trend in the United States, is using cheeses made with raw milk. We can look at some other notable outbreaks from the past decade. that one of the common factors in all of these is that there was no lethality step for these particular food products, or there might have been some cross-contamination with raw products. Across the board, we see produce, meat, flour-based ingredients, dairy, as well as nuts and spreads. Some of the more recent cases are going to be sprouts, including the one from this year with O103, several other sprouts at O21 and O26. Romaine lettuce and other baked kits have been associated multiple times over the last 10 years, not only with O157, but also with O145. Ground beef, bison, Lebanon bologna should be a processed type of meat product, but was also associated with some contamination with E. coli. Flour has had several different outbreaks, not only with flour by itself, but also in food products that were prepared for flour that were not necessarily cooked well before consuming. When we're looking at the cheeses, most of these are going to be either raw milk or they were cross-contaminated in the facility with raw milk. What are we going to do for controlling it then? And as I said earlier, it's not going to be good enough just to prevent the growth. What we have to do is kill it. As a result, juices that are going to be regulated under the juice HACCP is going to be looking at a 5-log reduction step. For the fresh produce, that we would suggest that you refer to the leafy green STEC action plan to find out what is going to be the best procedures in order to reduce your risks that are associated with fresh produce. For flour, part of this is going to really rely on trying to educate the consumer. This is an example of a picture that I took when I was making Christmas cookies a couple years ago, where the flour said, "Cook before sneaking a taste." And I'm of the generation that loved eating raw cookie dough. My roommates and I would make cookie dough for a snack and not necessarily bake it. So trying to get out of that habit of eating the raw cookie dough or raw scone doughs or things like that is something that we really need to advise the consumer. If you're a commercial company, such as Nestle's and General Mills, they have recently decided that they were going to use pasteurized flour for the cookie dough. So now you can go into the store where you'll see it saying it's safe to eat raw, you can eat directly, or you can bake it. Because once again, they know that they need to have a lethality step. They can't necessarily rely on the consumer to make that step, and therefore this is the change that they need to make. For meat products, once again, we're going to focus on the big 7. There are going to be the fact that these are going to be adulterants, and so we have to make sure that the intervention strategies on the carcasses are going to be such that will eliminate them. In the cases of fermented dried sausages, once again, we're looking at a 5-log reduction. We'll show you an example of when we were doing work with fermented dried sausages, that the acidity and the drying by itself is not going to be sufficient to get rid of E. coli 157 and all of its friends. But when you put it in conjunction with some mild heating, it will stress the system so that they will die off. For other kind of meat products, we need to make sure there's going to be validated kill steps. Once again, we're thinking about those that might have more of a mild kill step rather than those things that might be cooked to like 165. So what's going to happen with a rare cooked roast beef? For dairy, we're lucky if we're going to be able to make our food products with pasteurized milk. However, there are many growing number of individuals that really would like some of the raw milk cheeses. And we have documentation that a 60-day agent may not be sufficient to assure the safety of these food products if there was E. coli in that raw milk. So that acidity, that reduction in water activity, that drying process and aging process will eventually get the E. coli off, but it won't be necessary or won't be well for rapid inactivation. Therefore, what we're recommending is that there's some kind of heat treatment for the milk. Not necessarily a full pasteurization, but any kind of heat treatment would be useful. So stealing a slide from Kathy Knudsen is, if I control for Salmonella, do I control for pathogenic E. coli? And my answer is going to be the same. It really will depend. Here's an example of some work that we completed in the lab where we were comparing the thermal inactivation of several different pathogens in ready-to-eat meat products. And in this particular case, we were looking at Listeria, which is going to be the blue line, Salmonella, which is going to be the red line, and Shiga toxin-producing E. coli. Once again, we looked at all 7 serotypes that along with it, and that's going to be the green line. As you can see, we're looking for beef, deli-style turkey, as well as ham. In many of the cases, the green and the red, or the E. coli and the Salmonella, really had almost exactly the same thermal inactivation rate. And so in that particular case, yes, what controlled the Salmonella should control the E. coli. However, when we looked at the beef, we saw that there was going to be a divergence in that line, that the E. coli, the STEC survived longer at the lower temperatures in the beef than what the Salmonella did. Once they get up to higher temperatures, then they wouldn't be able to see that much of a difference. So in this particular case, no, you cannot use Salmonella as being your indicator for safety. However, across the board, if you would wanted to, you could use Listeria as being your indicator. So if you were able to get a 5-log reduction of Listeria, more than likely you had way more kill for the Aztecs. When we're trying to validate lethality, what should be your target? And as I said, there's going to be a couple of the food products such as the juice hassup and fermented dried sausages where we do have regulatory recommendations for 5-log reduction. This is based off of having some E. coli, probably around 10 to the 2 CFU per gram, plus adding in an additional 100-fold margin of safety. And we understand that people will be eating more than 1 gram of food product. So if you're looking at eating potentially 2 ounces or 3 ounces, you want to make sure that per serving, you don't have the E. coli 157 in there or the other Aztecs. As far as other foods, there aren't necessarily any clear guidance. And therefore, using that 5-log reduction may be prudent. If you're looking at a lower log reduction, then there should be some justification for that. So how do you go about doing the validation? Well, first of all, the best way of doing it is to actually get the food product, inoculating it with the high levels of pathogen, and then trying to find out what are going to be the inactivation kinetics. What's the difficulty that goes along with this is trying to replicate those conditions. You have to have a laboratory that's able to replicate that particular processing. It might have to do with what kind of relative humidity. Can you replicate the temperature profiles? It's not necessarily going to be an instant temperature up and instant temperature down. What kind of other dynamic conditions are there? Do you have any kind of surface drying in which there's going to be stabilization of the microbes that might be on the top? The other thing that is of concern is the fact that this is a low infectious dose pathogen, and we're concerned about working with very high concentrations of the pathogens, and we want to avoid any kind of lab-acquired infections. So once again, this is not necessarily something that you're going to do in your own processing facility, but we'll have to have a dedicated laboratory who are capable of doing this type of work. So if you can't do this in a laboratory, you really need that specific food processing equipment, then people want to do some surrogates. So then comes the pros and cons about using the non-pathogenic surrogates versus looking at other type of pathogen as indicators. If you are going to be using a surrogate, how close are the inactivation characteristics to your particular target? Gram-positive bacteria tend to be a little bit more heat resistant than gram-negative bacteria under most circumstances, but not necessarily under all. Salmonella is known to be extremely thermal resistant when it comes to low moisture, low water activity type of environments. We're also looking at, as our surrogate, does it share its acid tolerance? Does it share a salt tolerance? Once again, does it share a tolerance to drying? So what we're trying to do now is we'll take a look at what have been some of the validation data that is looking to see how you can inactivate E. coli 15787 in other pathogens in these particular food products. So let's take a look at an example is going to be cheese. Once again, there have been several outbreaks associated with a semi-soft cheese known as Gouda with E. coli. And if you get to a soft cheese, instead of having some inactivation, you might actually have growth. In the case of the soft cheese in France a few years ago, that was something that the E. coli actually could grow in that particular case. But if we're looking at a hard cheese, eventually the E. coli will die off. But there will be a long survival time in hard cheeses and semi-hard cheeses if they're made with raw milk. This figure is an example of a publication from about 10 years ago in which they took raw milk, specifically inoculated it with E. coli 157 at a very low rate of 10 colony-forming units per mil. But when you take a look at this figure, you see that the numbers are going to be closer to 100 per mil. Well, what happened is that during that first part of that fermentation process, you're going to have a little bit of growth that's going to get started until that acidity starts growing. The other thing is that as you're concentrating the curd and you're expelling the whey, now you're concentrating the microbes and you're concentrating them about a tenfold, which is what you're doing for concentrating the solids from the milk. So instead of having 10 colony forming units per gram, we're up to about 100 colony forming units per gram. Over time, we do have an inactivation. But we see that we do have survivals even by enrichment process all the way through 3 months. So in this particular case, even though we do have a rapid inactivation initially through that first 60 days, we still have infectious doses all the way through that shelf life. Therefore, what might be some recommendations? And we say, even if it's a mild heat step of milk, rather than a full pasteurization, we can potentially make these food products safe. So it's once again, we've got a lot more work that has been done, published in the literature about listeria. Can you use that listeria information to predict the inactivation of STECs in the milk? In the raw milk cheeses, what the recommendation is that there's about a 3-log kill of listeria in cheese milk that's used for soft cheeses. We know that gram-positive pathogens are typically more heat-resistant than gram-negative if they're in a higher water activity neutral pH environment. So therefore, is it possible that you can look at the data on the listeria and apply that to the E. coli 157 in a milk product, which would mimic high water activity neutral pH? This is some of the work that's still being in progress by one of the graduate students, Sarah Engstrom. And what she is doing is comparing the inactivation of Listeria monocytogenes, several different strains, along with the big 7 of the STEC in milk. And what we can see from here is the D values at various temperatures from 135 all the way to 150 degrees, is that if you're comparing the E. coli 0157 and all the rest of the STECs with Listeria, consistently we have shorter D values than what we do for the Listeria. So in this particular case, if you can get your 3-log reduction of Listeria in milk, the hope is that you're going to have more than a 3-log reduction of STECs. Therefore, if you do that heat treatment, say for instance that you're going to be heat treating your milk for 3 minutes at 140 degrees Fahrenheit, that may be more than sufficient to make that milk safe, but it won't necessarily be a full pasteurization, and you might still have a lot of that non-starter lactic acid bacteria that's there for flavor that will still be available. I mentioned also that the combination of acidity and low water activity alone may not necessarily be sufficient for dry fermented sausages. And we found that out by the outbreak that was in the early 1990s. In the late 1980s, however, we did some individual studies, and where first of all, we took a look at Listeria in dried fermented sausage, and we were very pleasantly surprised that within a fairly short period of time, we had at least a 4-log reduction of Listeria. When the pH got down to a 4.8, we had quite a bit of reduction, and then throughout the drying process, we were at the point where we're at detection limit. However, when we were looking at fermented dried sausages in E. coli 157, we had very little kill during this particular process. There was a couple of different things that were going on with this. Number one is that Listeria is more acid sensitive than what the E. coli 157 is. And the other thing is when we went back to the starter culture, we found out that starter culture actually had bacteriocin activity against Listeria, but it did not have any bacteriocin activity against the E. coli 157. So when we were finished with this particular study, we said if you've got a lot of E. coli in there, you might have still some left at the end of the time. Now, back in the late 1980s, we did not have a good understanding what the infectious dose was. And so we were caught by surprise when that first outbreak was associated with the dried fermented sausages, because we didn't realize that there would be just a few survivors and that could cause illness. Now, fast forward to 2010, and we needed to take a look to find out whether or not all the information that we had gathered with the mild heat treatments to add to kill the E. coli 157 in pepperoni or other dried fermented sausages could be applied to the rest of the STECs. So in this particular case, we made pepperoni from scratch. We purposely added the 0157 and all of its happy friends to the batter, stuffed it, did the fermentation, did the cooking, and did the drying for up to 20 days, and followed along the kill step. In this particular case, once again, we did have a lot of kill that went along with it, but we still had some survivors at the end of the kill or end of the cooking step, but we did have the rest of the kill was going to be throughout the rest of the time. What was set up that we did have about a 5-log reduction within 7 days of the heat treatment, but we also had some other E. coli that did survive all the way through 14 days. Then colonies were picked, trying to find out which were the predominant serotypes. All of our work was done on the 015787 as alternate processes. Is this still going to be the target? So if you can control 157, can you control the other STECs? And what we found out is that the 0103 was about equivalent or maybe a little bit less resistant to the whole process that 0157 was. So the good news was all that data that we had collected in the 1990s for thermal inactivation and that combination of mild heating and drying and acidity still would apply to the rest of our STECs. Now what we wanted to do is find out, well, why did those particular strains survive the most? Was it because they were more acid tolerant, or was it having to do with the drying tolerance? And in this particular case, what Chuck Casper did was he took a look at the individual strains to find out if any of them grew at in a laboratory media using hydrochloric acid down to a pH of a 4.8. So the hydrochloric acid is going to be less antimicrobial than what the lactic acid is, what we found in the pepperoni, but it still has the effect of pH. The take-home message of this is that all of these STECs were going to be acid tolerant in that all of them grew at a pH of a 4.8. However, the two strains that were the most acid tolerant were 0121 and 045. And they were actually slightly more acid resistant than 0157 in a laboratory media. However, notice that these were not the ones that showed up in the pepperoni. However, when we took a look at what the effect of the salt tolerance was, and this was, once again, making laboratory media, the water activity was adjusted to about a 0.92, which would be similar to what the water activity would be in the sausage using sodium chloride. It ended up being about a 4% salt solution. And what we found on this particular case is that 0157 and 0103 were more salt tolerant. They survived for longer periods of time than the rest of the serotypes. The other thing that was a take-home message was the higher the temperature, the more rapid inactivation. So in other words, it was stressful enough that it helped beat them down. So these might help with the explanation of why these particular strains were more resistant to that process in the pepperoni that it had actually to do just as much with the salt tolerance as it did to the acid tolerance. Now, as we're looking through other kinds of validation, once again, we're thinking about, well, what can you do potentially in your own processing facility? And we say what's really important is that if you're going to choose some surrogates, that you do consider its tolerance for acid, what it can survive or grow at lower pH values, what is going to be the effect with water activity, and what's going to be its effect in in heat with high humidity versus low humidity. Now, there are definitely some surrogates that are either have been validated or in validation. And what you can use is you can look at the FSIS website. There are 5 ATCC strains that are used as surrogate E. coli that are used for select antimicrobial treatments for carcass interventions. They could also potentially be used for certain kinds of cooking, fermentation, freezing situations. And then there was also a study published several years ago using a lactic acid bacteria starter culture that was validated as a surrogate for Salmonella and E. coli 157 in ground form jerky. This is the table that came from that publication in 2009. And what they were doing was looking for at least a 5-log reduction of E. coli 157 as well as Salmonella. And they looked at 2 different types of starter cultures. Both of them were pediococcii, but from different sources, and demonstrated that with these various types of processes, and you can go to the publication to look at what those processes are, consistently, the 2 surrogates had less kill than what the E. coli was and the Salmonella. In other words, they were good surrogates. So if you have at least a 5-log reduction with these particular bugs, you should have at least a 5-log reduction of the E. coli 0157 or the other STECs and the Salmonella. Now, once again, this was validated for under various circumstances, but this is still for one particular product line, in being that it's going to be a jerky product. So in summary, the STECs, the 0157, as well as all their friends and other pathogenic E. coli are definitely inevitable contaminants in raw commodities. You can't get away from them. Therefore, as we're looking into the future, we should be looking at other kinds of foods that might be affected. So even though this may not necessarily be an emerging pathogen, we may have emerging issues. The best control strategy is to reduce or eliminate its presence in food. And we definitely need more research to identify viable intervention strategies for fresh produce. How are we going to kill it on our romaine lettuce or on our sprout seeds? For other types of food products, we don't necessarily have to go to a full cook. What we can do is use a sub-pasteurization, but use it in combination with other types of hurdles, such as acid or salt or reduced water activity. And this could help stress the system enough so that they will die off, so that they are no longer infectious. Other kind of lethal processes should be validated to determine efficacy. And if you are going to choose some surrogates or other indicators, it must be very carefully chosen for a particular application. There's a lot of work that's already been published, but there's still a lot of work that has to be completed. So with that, we'll look for any kind of questions, and Alan Saylor will be your main contact from here. Thank you.